The following video will demonstrate how to use the Ovarian Kaleidoscope database, also known as OKdb. This is a database we set up more than 10 years ago together with a group of Stanford undergraduate students. We put in published literature on ovarian genes. After the initial submission of several hundred genes, we continue to submit new genes during the last 10 years, and now the database as January 2012 has more than 3,500 genes. The database has been supported by NICHD, the SCPIR Center, SCCPIR Center, and especially with the help of support from Dr. Lou DiPaolo. The following videos will continue to demonstrate two parts. The first part is how to do the text-based searches. The second part will deal with how to search DNA microarray data. If you go to the front page of OKdb, you can see a very simple gene name search. And for example, you just type in caspase. You can see there are 14 genes that has the name caspase in there. And then let's just go to caspase 3, the first one. And for each gene page, you can see we have all the different names of the particular gene. We have on the right-hand side the human chromosome localization. And you can go to other databases such as Entrace gene page by simply clicking on the Entrace gene. And in the Entrace gene page, it has a lot of different information, and you can go into the detail. When you go back to the caspase 3 database, you can see that you can also go to MGI, which takes a little bit longer to load, just because the nature of the database. Once you go to MGI, you can see the mouse genetic, and in this case, it asks you whether this is a caspase 3. You click on this caspase 3 on the MGI database, then you have data that really deal with the mouse, especially a lot of the information on expression and transgenic animals. That's all available there. So now let's go back to OKdb now. And you can see in every OKdb gene page, you can see a general comment section describing what the gene is all about, and then followed by general function, which is such as cell death and survival, apoptosis, and this is particular one is an enzyme. And you can see cellular localization, where this gene is localized, and ovarian function. This is the most important part, which is only found in the ovary database, on our database. And it says expression of caspase 3 detected in follicular cell of attractive follicles. And you can click on each of this blue link, and you can go to the original data, original publication. And from there, you sometimes can go to the full text literature. And we also talk about the expression regulation. In this case, this gene is regulated by LH and FSH. And all the comments, you follow the comment, you can see the data that the publication that support this LH and FSH regulation. Furthermore, we look at the ovarian localization. This gene is expressed in oocyte, granulosa cell, Zika cell, luteal cell, and surface epithelium. And the different follicular stages and interesting aspect is the mutation. For example, this one talk about the mutation in the mouse, and this is original literature. For every gene pages, for every particular gene page, you can actually use this discus. You can log into this, and you can put in any comment you want, and that will be shared in the future for people who are interested in this particular gene. The most important aspect of the OKdb searches is something, as I mentioned, Google cannot do. Like, for example, you can search all the gene in the ovary that's expressed in the oocyte. You do simple click on the oocyte. And the same criteria can be used certainly to search other things, such as the ovarian specific biological function. You want to see all the genes that's involved in ovulation, for example. But let's go back to the oocyte. So you do the search. This is oocyte genes in the ovary that has literature 1,000 some genes, and that certainly is too much for you to try to figure out. But on the other hand, we have a combined search features. That is, you want to see oocyte genes that also is a ligand. So that means oocyte secreted genes, oocyte secreted hormones, cytokines, or growth factors. You do this combined search now, you suddenly have only 72 genes now. And we have another feature. Please remember, now all the search is continuous because whatever we put in there are still in there. The oocyte is still in there, ligand is still in there, and you have a cell-specific gene. That is, this gene is only expressed in oocyte and not in other, such as granulosa cell or in the ovary. If you do even more specific search now, the ligand that's only secreted by oocyte become 30. And this will become very interesting. These are the genes the oocyte secreted to regulate, likely to regulate the granulosa cell functions. And certainly you can click on each one of the gene and you can go to the original data, the gene page and the original data. And if you want to start a new different search, very important, you remember to do clear forms. Now I clear all the form. You see that the oocyte disappeared now. And I can do some other searches. For example, I want to search the gene that has genetic changes. For example, I want to see female fertility status. I want to see infertile ovarian defect. Due to ovary gene defect, this gene defect in the ovary will cause infertility phenotype. You click on this here, you can see, wow, 180 genes. Certainly that's too much to do. But again, you can zoom in by doing the sequential searches. And you say, well, maybe I will do this search, but I only want the species. That is, I only want to look at human. The human gene that's give you infertility phenotype, and that's ovary defect. You can see that now it's only 18 genes. So these are very useful clinical data to the clinicians, and certainly to combine this data with the basic science literature will make this finding very interesting. You can also say, well, this 18 genes, I will only want to concentrate on genes that's expressed in the nucleus. So we have another searches here. This is again combined search. You can say I only want mitochondria or only want to look at secreted ones. So let's look at human genes, infertility genes that's found only in the expressed only in the nucleus. So now you're zooming into 7 genes now. So with this type of approach, you really can look at ovarian genes in a kaleidoscopic way. That is, you from different angle, just like you're playing with kaleidoscope. From different angle, you look at the ovary. You can get totally different but interesting results. Okay, so this search, and always remember to clear the form. This type of search, you have to do it yourself, and then you begin to find new things which even the frequent users may not even know, and it will fit your own interest to understand a particular gene. So now I want to show you other features that's unique for the OKdb. For example, we now put in a gene. This one I know, BDNF. It's a ligand gene. It's a hormone. Okay, so brain-derived neurotrophic factor. I want to show this gene to you because we not only have individual genes, we are beginning to show the relationship between the genes. In this particular case, you can see this gene, every gene has a receptor ligand interaction line. So if you click on the receptor ligand interaction, it tells you this BDNF actually binds to its receptor, which is called NTRK2. And this talk about its receptor. And this gene is expressed in the ovary because OKdb has a link. And it also tell you where it's localized. It's found in the oocyte and granulosa cell. And as you already know, the BDNF, we already done the search, and in the same page it tell you the BDNF is found in the cumulus cell and granulosa cell. And if you click on this link, you can certainly see the receptor gene in terms of the literature. Okay, and now go back to BDNF. I want to show you why this database is interesting. We have DNA microarray data embedded for every gene. So if you click on Show Data, we show you several different types of data. One is oocyte and early embryo data. We have 4 different data sets, DNA microarray data sets, talking about oocyte, pre-implantation, and other the human and mouse development. The default one that shows up is always this one, which is a pre-implantation mouse embryo. And you can see it has the original literature. If you click on description, you can go to this PMID and find the original publication. And the field, it tells you what are the fields that's in here, shown in our plotting. And our field shows GB stage, M2 oocyte, zygote, early 2 cells. This is very extensive DNA microarray data. And you can put your mouse cursor on here. You can see BDNF is expressed in all these different stages in the zygote, for example, or in the 4-cell stage. For all the stages, this gene is expressed. But the most interesting data I actually want to show you for BDNF is in the second section. We talk about follicle maturation and ovulation array. And certainly you can also look at embryonic ovary and other array if you're interested. So if you do this click, you can see that this is the follicle maturation and ovulation array, a very extensive array. The immature mice were treated with PMSG or ECG at time 0, and you can see this is the second one is 2 hours later, and this continues until the 48 hour. And 48 hours here. You see that? There is an initial increase after FSA treatment or ECG treatment, and then it goes down. However, from this point on, the 49 point and later is every hour. This is very detailed. 12 hours after HCG stimulation, you can see the ovulation LH-HCG induce a major increase in BDNF. And this data allow us to actually study the effect of BDNF as a factor which regulates the oocyte function, because the receptor, as I just mentioned to you, is found in oocyte. And this is the paper, if you're interested, looking at Kawamura paper and published in PNAS, showing that indeed, if you look at this figure, this is the DNA microarray data set, and then we confirm it using real-time RT-PCR. This is the receptor DNA microarray data set, and this later on is found is very high in the oocyte. This is the granulosa cell producer factor that regulate the granulosa cell that regulate the oocyte function. So Ovarian ddNF promote the development of oocyte into pre-implantation embryos. So if we go back to the DNA microarray, and we not only have this oocyte early embryo array and ovary array. For people who are interested in testes or endometrium or even pituitary, we also have the same thing. So the idea is to plot all this data for the user so that it's very convenient to see the regulation. And give you another example of this, the value of this database is if you look at FOSS, the ovulation array show you the most striking data. That is, after the LH-HCG search is a major increase. And these are the early induction genes, very important for the function of hormones, to mediate the function of hormones. For all the genes, you can actually have another link, which is called the BioGPS. If you click on BioGPS, this is another database which we link to them. And in the BioGPS, it shows all the genes in the human body, and so it's loading right now, and it will show all the different 50-some tissues in terms of the expression of the particular gene of interest. If you're interested in ovary specificity of the genes, of certain genes, then you can certainly search this database, link to this database. Okay, so if we go back to the search page, there's another very important feature. It's called a KEGG. KEGG is a website in Kyoto Encyclopedia of Gene and Genome. The loading of this database, because it's too big, so it takes a few minutes, so please be patient. What happened here is, in this database, they have put on 185 maps, and for a lot of different cell pathways that's used, for example, for adhesion junctions. It's used by every cell in every different tissue, organ in the body. Even some of the adipocyte cytokine signaling pathway, all the cytokines probably are used by different tissues. So you can go to this database, and for example, you look at apoptosis. Apoptosis for sure is every tissue uses apoptosis. And these are all the genes listed here are the genes that's already in our database. And if you click down here to view the ovarian genes, and all the genes will be marked in red color. If you click on this, and you can see this is the whole pathway. All this pathway and all the red ones are already studied in the ovary. So if you look at the one that's not red in color, you know they are the missing genes. They are almost sure to play important role in the ovary. So this is another way of looking at the ovarian genes as a group now. So now we go back to the search, and for clinical importance, we also put in the POF genes. So those are all the literature that has premature ovarian failure, and you can see that there are 43 genes, and certainly new genes are being added all the time. And another one is if you have to clear the form again, and you look at the PCO genes. And PCO genes, there are 55 genes, and certainly each of these genes, you can click on it, and then you can see the description of the gene in the mutation section. Human natural occurring substrate type, and these are the gene talking about why, these are the data talking about why this gene is related to the PCO story. So I hope this presentation is helpful and allows at least the beginning part of how to use this database. and good luck with your research.